Hello and welcome students. Today we will discuss about immunotolerance. So let's have a brief introduction. An understanding of the immune response as a complete physiologic system requires some understanding of the off signals in addition to an understanding of the on signals. One of the key mechanisms for controlling the lymphocyte response to antigen is by the induction of tolerance called acquired immunological tolerance rather than activation. Tolerance can be induced or tolerization in all of the cells of the immune system, including T cells, also known as T lymphocytes, the antibody forming B cells, also known as B lymphocytes, and natural killer cells. Tolerance can be achieved anytime in the ontogeny of the cell, provided it expresses a receptor for the antigen. We refer to tolerance induced during the early stages of lymphocyte development as central tolerance, and to tolerance induced in mature lymphocyte as peripheral tolerance. There are two major consequences of lymphocyte inactivation: deletion, in which exposure to antigen results in the elimination of cells by apoptosis, or alternatively energy, in which exposure to antigen results in an unresponsive or inactivated state. These mechanisms are not mutually exclusive, as cells that are initially energized by exposure to antigen may be deleted at a later time. In the following sections, we will discuss how both immature and mature lymphocytes can be made tolerant to antigen. So now let's come to the details of immunological tolerance. Immunological tolerance to different self-antigens may be induced when developing lymphocytes encounter these antigens in the generative or central lymphoid organs, a process called central tolerance, or when mature lymphocytes encounter self-antigens in peripheral, secondary lymphoid organs or peripheral tissues called peripheral tolerance. Central tolerance is a mechanism of tolerance only to self-antigens that are present in the generative lymphoid organs, namely the bone marrow and the thymus. Tolerance to self-antigens that are not present in these organs must be induced and maintained by peripheral mechanisms. With this brief background, we proceed to a discussion of the mechanisms of immunological tolerance. Tolerance in T cells, particularly CD4+ helper T lymphocyte, is discussed first because many of the mechanisms of self-tolerance were defined by studies of these cells. In addition, CD4+ helper T cells orchestrate virtually all immune responses to protein antigens. So, tolerance in these cells may be enough to prevent both cell-mediated and humoral immune responses against self-proteins. Conversely, failure of tolerance in helper T cells may result in autoimmunity manifested by T cell mediated attack against self-antigens or by the production of autoantibodies against self-proteins. Now let's come to the central T lymphocyte tolerance. The principal mechanisms of central tolerance in T cells are that of immature T cells and the generation of CD4+ regulatory T cells. The lymphocytes that develop in the thymus consist of cells with receptors capable of recognizing many antigens, both self and foreign. If a lymphocyte that has not completed its maturation interacts strongly with a self-antigen displayed as a peptide bound to a self-major histocompatibility complex MHC molecule, that lymphocyte receives signals that trigger apoptosis. Thus, the self-reactive cell dies before it can become functionally competent. This process, called negative selection, is a major mechanism of central tolerance. The process of negative selection affects self-reactive CD4 plus T cells and CD8 plus T cells, which recognize self-peptides, displayed by class II MHC and class I MHC molecules, respectively. Negative selection is imperfect and numerous self-reactive lymphocytes are present in healthy individuals. As discussed next, peripheral mechanisms may prevent the activation of these lymphocytes. Now let's come to the peripheral T lymphocyte tolerance. Peripheral tolerance is induced when mature T cells recognize self-antigens in peripheral tissues, leading to functional inactivation, that is energy or death, or when the self-reactive lymphocytes are suppressed by regulatory T cells. Naive T lymphocytes need at least two signals to induce their proliferation and differentiation into effector and memory cells. Signal 1 is always antigen, and signal 2 is provided by co-stimulators that are expressed on antigen-presenting cells APCs, typically as part of the innate immune response to microbes or to damaged host cells. It is believed that dendritic cells in normal uninfected tissues and peripheral lymphoid organs are in a resting or immature state, in which they express little or no co-stimulators such as B7 proteins. These dendritic cells may constantly process and display the self-antigens that are present in the tissues. T lymphocytes with receptors for the self-antigens are able to recognize the antigens and thus receive signals from their antigen receptors, signal 1. But the T cells do not receive strong co-stimulation because there is no accompanying innate immune response. The presence or absence of co-stimulation is a major factor determining whether T cells are activated or tolerized. Now come to the energy. Energy in T cells refers to long-lived functional unresponsiveness that is induced when these cells recognize self-antigens. Self-antigens are normally displayed with low level of co-stimulators. Co-stimulation is thought to be the basis of energy. Energetic cells survive but are incapable of responding to the antigen. The two best defined mechanisms responsible for the induction of energy are abnormal signaling by the TCR complex and the delivery of inhibitory signals from receptors other than the TCR complex. When T cells recognize antigens without co-stimulation, the TCR complex may lose its ability to transmit activating signals. In some cases, this is related to the activation of enzymes, ubiquitin ligases, that modifying signaling proteins and target them for intracellular destruction by proteases. On recognition of self-antigens, T cells also may preferentially engage one of the inhibitory receptors of the CD28 family, cytotoxic T lymphocyte, associated antigen 4, CTLA4 or CD152, or program that protein 1, PD1. Energetic T cells may express higher levels of these inhibitory receptors, which will inhibit responses to subsequent antigen recognition. So now let's come to the deletion, apoptosis of mature lymphocytes. Recognition of self-antigens may trigger pathways of apoptosis that result in elimination, that is deletion, of the self-reactive lymphocytes. There are two likely mechanisms of that of mature T lymphocytes induced by self-antigens. Antigen recognition induces the production of pro-apoptotic proteins in T cells that induce cell death by causing mitochondrial proteins to leak out and activate caspases, cytosolic enzymes that induce apoptosis. In normal immune responses, the activity of these pro-apoptotic proteins is counter-reacted by anti-apoptotic proteins that are induced by co-stimulation and by growth factors produced during the responses. However, self-antigens, which are recognized without strong co-stimulation, do not stimulate production of anti-apoptotic proteins. The relative deficiency of survival signals induces that of the cells that recognize these antigens. Now let's come to deletion of T lymphocyte. Recognition of self-antigens may lead to the co-expression of that receptors and their ligands. This ligand-receptor interaction generates signals through the that receptor that culminate in the activation of caspases and apoptosis. The best defined that receptor-ligand pair involved in self-tolerance is a protein called FasCD95, which is expressed on many cell types, and FasL, which is expressed mainly on the activated T cells. The Fas pathway may also be involved in that of some B cells in germinal centers. Coming to the B-Lymphocyte tolerance. Self-polysaccharides, lipids and nucleic acids are T-independent antigens that are not recognized by T cells. These antigens must induce tolerance in B lymphocytes to prevent autoantibody production. Self-proteins may not elicit autoantibody responses because of tolerance in helper T cells and in B cells. It is suspected that diseases associated with autoantibody production, such as systemic lupus erythematosus, are caused by defective tolerance in both B lymphocytes and helper T cells. Now let's see about central B cell tolerance. When immature B lymphocytes interact strongly with self-antigens in the bone marrow, the B cells either change the receptor specificity, that is receptor editing, or are killed, that is deletion. Receptor editing. Some immature B cells that recognize self-antigens in the bone marrow may re-express RAG genes, resume immunoglobulin Ig light chain gene recombination, and express a new Ig light chain. This new light chain associates with the previously expressed Ig heavy chain to produce a new antigen receptor that may no longer be specific for the self-antigen. This process of changing receptor specificity, called receptor editing, reduces the chance that potentially harmful self-reactive B cells will leave the marrow deletion. If editing fails, immature B cells that strongly recognize self-antigens receive that signals and die by apoptosis. This process of deletion is similar to the negative selection of immature T lymphocytes. As in the T cell compartment, negative selection of B cells eliminates lymphocytes with high affinity receptors for abundant and usually widely expressed cell membrane or soluble self-antigens. Energy. Some self-antigens such as soluble proteins may be recognized in the bone marrow with low avidity. B cells specific for these antigens survive, but antigen receptor expression is reduced and the cells become functionally unresponsive, that is anergic. Now let's see peripheral B cell tolerance. Mature B lymphocytes that encounter self-antigens in peripheral lymphoid tissues become incapable of responding to that antigen. According to one hypothesis, if B cells recognize an antigen and do not receive T cell help because helper T cells have been eliminated or are tolerant, the B cells become anergic because of a block in signaling from the antigen receptor. Anergic B cells may leave lymphoid follicles and are subsequently excluded from the follicles. These excluded B cells may die because they do not receive necessary survival stimuli. B cells that recognize self-antigens in the periphery may also undergo apoptosis, or inhibitory receptors on the B cells may be engaged, thus preventing activation. As mentioned earlier, regulatory T cells may also contribute to B cell tolerance. Now coming to the conclusion. Many strategies for inducing immunological tolerance involve reproducing the mechanisms involved in natural central tolerance, the phenomenon by which self-tolerance is maintained among immature lymphocytes developing in the central lymphoid organs. For developing T cells, tolerance occurs in the thymus, the central organ for the T cell development. For B cells, development occurs in the bone marrow, and an encounter with self-antigens can induce tolerance there among the immature cells. Despite improvements in immunosuppressive drug therapy, teaching the immune system to regard a set of foreign antigens presented by the organ graft as self, that is tolerance induction, has become an important goal for several reasons, as it would eliminate the need of chronic immunosuppressive therapy, which is associated with lifelong increased risk of infection and malignancy and other side effects.